So let's build on the basic cardiac anatomy that we've learned so far this week. We are learning basic structure and features of the heart, and we're going to build on that by adding the vessels. The heart connects to the vasculature to pump blood throughout the body. And then next week, we will build on the basic structure with some more advanced structure, and then now start to include function, the physiology, how this structure dictates function. That is, how the heart and how the vessels work. So the vessels proper, the vessels themselves, have generally 3 layers, and this is now old news. Most of you have seen this in lab. If you weren't too distracted by the dissection of the heart and you went through the rest of the lab and you paid attention and took notes, you'll know there are 3 layers: the tunica interna, or the tunica intima, which is the extension of the endocardium. Remember that single layer of epithelial cells that lines the entire inside of the vascular system? This is the innermost layer and the continuation of the endocardium. Next, in the middle, the tunica media, where we find potentially some elastic fibers, but where muscle, smooth muscle, will reside. And in this case, they're both rather thin. These are only to illustrate the layers. And lastly, the thick, dark blue layer on the outside is the tunica externa, the outermost layer surrounding the 3 layers, or surrounding the internal 2 layers of any given blood vessel. And so this is meant to be a nondescript generalization. These 3 layers are always apparent, but in different proportions. And you might know that this is a vein. We're not going so far as to name it now, but I will contrast the differences between the major categories of blood vessels in the cardiovascular system. So if we group them roughly, you have arteries and veins. And anywhere that you land within the cardiovascular system, other than the heart, the tube, the vessel that you're in can be grouped into one of those 2 categories. And the key structural differences that differentiate these types of vessels are, first and foremost, the arteries have elastic properties. The internal and external elastic lamina make the artery springy. And you saw an example of that in lab, how resilient and bouncy and springy the aorta was for your sheep specimen. Similarly, in humans, arteries are springy. They're resilient. They want to return to their original shape. And that is a really important characteristic for understanding their proper function. They want to return to their original shape. They are resilient. The class of vessels, arteries, also tend to have more smooth muscle. In very general terms, an artery has more smooth muscle than a vein, and this allows control over the diameter of that vessel. So we can contract to make the vessel smaller, make the diameter smaller and more narrow. We can relax, which will open up the vessel, all in an attempt to regulate pressure and flow, which is coming in a week or 2's time. But we can constrict the contents that are in that vessel using the smooth muscle in the tunica media. So arteries have elastic properties. They have more smooth muscle. You can see that contrast with the corresponding layers on the right-hand side and veins over here. Very thin. The walls in general are very thin. They might be 1/10 of the diameter in total in a vein. They're really floppy. They don't have a lot of structure. They can be deformed, but they don't want to spring back to their original structure. They do have something unique that arteries don't, and that's obvious looking in the middle of the vein here, these one-way valves, a lot like semilunar valves, if you think about it, that prevent the backflow of blood. And this is important because pressure on that side is relatively lower, the blood moves through more slowly, and if we have to get blood all the way back up to the heart from the feet, without pressure to do that, without blood moving at sufficient speed to do that, this system allows the blood to ratchet up in increments. The valves will stop the blood from falling back down to the feet and pooling in the legs. Which is really important if you want to stay conscious or stand around, walk around, lecture to a class. Now, between these two broad categories are a somewhat special grouping of vessels called the capillaries, which are purpose-built for exchange of nutrients, exchange of molecules across the surface. They're very, very thin-walled. They have a single layer of epithelial cells and a basement membrane, and that's it. So it's really easy for things to get into and out of capillaries. You can't see them with the naked eye. So we have arteries on one hand and veins on the other. We know some distinguishing features that will separate the two, and in the middle are capillaries. If you were to trace the path from the heart through a full circuit back to the heart, you would progress through the vessels in this order. From the heart, you would move first to the aorta, the single largest artery in the body. And then off of the aorta, you have a number of branches, and these are all arteries. Not as large as the aorta, still have elastic tissue, still have some smooth muscle. And they are larger than arterioles, which are the next smaller set of branches that you would encounter. And the arterioles are really where the walls get meaty. There's a lot of muscle in the arterioles. That's where we have a lot of capacity to constrict or relax, to change the diameter of the vessels. Now those divide and continue to divide, and you end up in such small, numerous vessels, the capillaries, single cell-walled capillaries that allow exchange of molecules across their surface. As you pass through the capillaries, instead of this really diffuse, spread out network of vessels, they start to collect. Capillaries merge to form venules. They're really small veins. Venules in turn merge to form veins, and then veins merge to form two vena cava. Only two in the entire body. All of the venous vessels on the return side share similar characteristics. There's little variation. They all have very little muscle, very thin walls, are very floppy. There are some veins that are muscular, but it doesn't really provide much constriction. This is the order in which you would encounter the vessels in the body. So arteries. I probably have given you all of this information already, but we already know arteries carry blood away. The pulmonary artery carries blood away, even though it's deoxygenated blood. We know the walls are elastic, and that's it's purposeful deliberately so that the walls want to return to their original shape. We'll see how the arteries store pressure and rebound in response to pressure. Maintaining that shape allows them to propagate or distribute blood distally throughout the arteries and arterioles in the body. Then, as mentioned before, the other defining characteristic is the muscle in the tunica media. to constrict the vasculature, make the space smaller, would increase pressure. And without pressure, there is no flow. So if you can constrict and increase pressure, you push blood through those vessels much more quickly. The velocity goes up, pressure is maintained, and tissues are perfused. Without pressure, there is no flow, and it's because of this smooth muscle that we are able to regulate pressure. Much more on pressure coming up. We're introducing pressure as an idea. A lot more detail incoming, don't worry. Let's briefly explore the idea of these arteries expanding and recoiling and acting as pressure reservoirs. So we'll use the aorta as our candidate artery. It's easy to observe. We know where it is, what it looks like. The first large artery that we run into when we leave the heart. And blood only leaves the heart when the ventricle contracts. So as the ventricle contracts, there's less space in the ventricle, blood is expelled. Where is it pushed to? Into the aorta. Now there's already blood in the aorta. It's not empty. So you're trying to cram another beat's worth of blood into an already full, limited space. So what happens? You either push everything through or the space accommodates the incoming blood. And because the aorta is elastic, and arteries in general are elastic, it expands to accommodate the incoming blood. So we see that shown here with the arrows pointing outwards. As blood is injected into the aorta, there's very little room, so it stretches to make room. Now, just from the fact that the heart has contracted, has squeezed blood in, we get movement. Blood moves through the arteries because more has come into the artery. We're pushing blood through the aorta. But this isn't the only reason that we get flow through the aorta. The heart contracts and pushes blood through directly. But as soon as that initial stimulus, that push, is gone, we're left with a situation where the aorta has stretched, the pulmonary valve might now be closed, there's no more blood coming in, and it wants to recoil. It wants to return to its original shape. When it does that, it squeezes the blood in that confined space again. Pressure goes up, and it looks for an escape route. The only escape route is to continue along the arterial pathway through the rest of the body. You can see that squeezing highlighted here. There's nothing mechanically squeezing the aorta. The natural properties of the elastic tissue in the walls return it to its original shape. And so for that reason, blood will continue to flow through the arteries, through the arterioles, towards the capillaries. So two-pronged approach. Blood is pushed by the heart as it squeezes, and there's recoil by the aorta and the arteries as there's this reflex wave as the walls return to their original shape and push blood through the arterial system. So it's the aorta and arteries that generally are responsible for this recoiling and this rebounding response to conduct blood further into the arterial system. And we would call those elastic arteries. They are larger, more elastic. They still have some musculature, but it's not the defining feature of these larger arteries. The elastic fibers are what causes them to recoil and push blood further into the arterial system. And for that reason, we say they act as pressure reservoirs. They store up the potential energy of the heartbeat pushing blood into the arterial system. Then we reach another class of arteries. If the large arteries and the aorta are the pressure reservoirs and the elastic tight arteries in the arterial side, the muscular arteries are what we would call distributing arteries. That is, they have the ability to constrict, to maintain pressure, like we talked about, and they can use that constriction to divert blood to where it needs to go. Let's say you're exercising. Muscles need blood. Other tissues, maybe like your stomach, don't. And so these distributing arteries, the muscular arteries, might really constrict the arterioles that go to the stomach so blood doesn't go there, or not as much blood goes there. And they might relax so that more blood goes to the muscles that are exercising. They can divert the blood accordingly. They can pick the direction of blood to flow to the tissues that need it. So these are two general classes: aorta and arteries as conducting arteries, and then the arterioles as distributing arteries. Another way to think about the types of arteries on the arterial side. Now, most arteries will branch until they reach capillaries. In some cases, though, you will observe, and you see this more on the venous side, you will observe arteries that merge. And that merging is called an anastomosis. One anastomosis, multiple anastomoses. So an anastomosis is the union of two vessels. It's relatively rare on the arterial side. It's much more common on the venous side. But it does happen, especially in places where you need redundant blood flow. What happens if one of the arteries gets blocked? If there's only one supply conduit and it's blocked, whatever it's supplying doesn't get oxygen, doesn't get glucose, doesn't get hormones, doesn't get anything. So these anastomoses provide an alternative route for blood flow. And we'll see these in the brain, the heart, which are both critical organs for survival. They have multiple redundant pathways. They'll have multiple arteries perfusing the same area of tissue, or you'll see them merge so there's an alternate route for blood to flow. The rest of the arteries that don't merge could be called end arteries. And in these cases, if that artery itself is blocked, then all the distal tissues are left wanting. They don't get any fresh blood, they don't get oxygen, they don't get nutrients. So sometimes having that redundancy is important, and you'll see that in the primarily important organs of the body. Now the flip side to this, if instead of merging an artery branches, which is very common on the arterial side, we'd call that a bifurcation, the division of any one artery into two or more arterial branches. This is much more common on the arterial side, especially as you get into smaller and smaller vessels. You can think of these as branches on a tree or levels of branching. there's an exponential division of the arteries on the arterial side, so that the total volume of arteries is massive as you approach the capillaries, and I'll show you that coming up. Now, we already talked about this. On the other hand, other than arteries, there are the veins, which are generally uninteresting. They are floppy, they are thin-walled, and they have characteristic one-way valves that catch blood and allow it to return to the heart under low pressures. You've seen some of these valves in lab if you did the demonstration that Matt talked about. I can't stomach doing that personally, so I've seen it in theory, or I've imagined it. Much thinner walls, and you can see that highlighted here. Only 1/10 of the diameter is made up by vessel wall. They're very loose, floppy. They don't maintain an original rigid structure. So notice the cusps here, these pockets, there's the head-on view at the top on the right-hand side, much like the semilunar arteries, but they don't entirely, they don't have 3 cusps like the semilunar arteries do. So the large veins returning to the vena cava are only formed from the anastomosis of multiple venules. Veins are formed from the union of several venules. The venules are just small veins, and they in turn are formed by the union of several capillaries. They have very similar function and structures, and they serve only to transfer blood from the capillaries into the veins proper. Now, I wanted to show you a really interesting video about capillary exchange, but that will have to hold off until next class. I will instead just show you briefly. I'll put this into context. You know, because you've heard it, or because I've said it, that the capillaries are small, but seeing singular red blood cells line up in a row spanning the entire diameter of the capillary, I think really makes that idea hit home. They are so small that only one red blood cell can fit through a capillary at a time. And this is the reason that blood doping or administering EPO is dangerous, in that erythropoiesis, making more new red blood cells, overcrowds the capillaries. Instead of them lining up one at a time in sequence to go through the capillaries in order, all of these new red blood cells clamor to get into the capillary at one given point in time. And so instead of there being flow, the capillaries get jammed and no flow occurs. No red blood cells go through, no oxygen is delivered, no CO2 is removed, and the person, if the red blood cell count is high enough, will die. We'll touch more on that later. You can see a nice EM scan on the bottom right. Video coming on Tuesday. Have a wonderful weekend. Sign up for Turning Point.